I'm Professor Tommy Borman and I manage the Shallow Marine and Coastal Research Infrastructure as well as the fully integrated Sino-Wandle Coastal Node here from the Ocean Sciences campus of the Nelson Mandela University in Gqeberha. The Shallow Marine and Coastal Research Infrastructure concept was well developed by the scientific community as well as coastal managers through several reports produced over the past 2 decades. The timing of SMCRI was also very fortuitous, as several government departments with a strong history in coastal research changed their business model to one of more service delivery, which left a major gap for the provision of coastal research infrastructure and data holdings in South Africa. Despite the concept being well developed, I held workshops with 350 coastal scientists from 30 research entities within South Africa, and we further improved the SMCRI concept to come up with 15 research platforms that would be distributed around the coast. Luckily for SMCRI, 2 business units of the National Research Foundation, namely SCION and SIAB, had already established some of these platforms. So SMCRI was able to quickly get off the ground through targeted supply chain management. We procured all or most of the large research infrastructures, such as the aircraft, the hyperbaric chamber, the large coastal craft, and as well as numerous instruments for the biogeochemistry laboratory. SMCRI was very successfully launched in 2018, and then of course the COVID-19 pandemic struck, and COVID had a major impact on SMCRI. So it took nearly 2 years, and we're only really starting to get out of the impact of COVID now, and we're well on track to get our operations, our procurement and filling of all the vacancies up and going now. But it does mean that now, after 5 years, we are looking at completing our rollout of the research infrastructure only in basically year 6. But it's not all doom and gloom. We used our time very effectively over the last couple of years. We seconded 7 staff members from SIAB and SCION. We employed 15 excellent staff members, and together we've made a significant impact in the national system of innovation through the publication of more than 92 manuscripts in peer-reviewed journals with a mean impact factor of more than 4. We also had a major impact in human capacity development. More than 130 postgraduate students have been trained by the SMCRI, either the staff or through the use of our research platforms, and the majority of these 130 students are from disadvantaged communities and all come from historically black universities. Our science engagement team also did an excellent job to reach more than 2,041 learners, 168 educators, as well as 968 members of the public over the last 5 years. Our platforms attracted more than 180 coastal scientists from South Africa, the region and globally that made use of our research platforms and data holdings. And our biogeochemistry platform managed to analyze more than 13,000 samples over the last 3 or 4 years that the platform has been in operation. Our databases, our websites attracted more than 6,600 unique new users. Most of our platforms benefited society in some way or another. So despite all the challenges that we faced over the last 5 years, SMCRI has been very successful in reaching the majority of its objectives, and I think we are well on track to complete our rollout in the next year, and we really look forward to the next 10 years.
My name is Nozi Hamaze. I am a Science Engagement Officer. Through SMCRI, we were able to engage with many people, ranging from learners, educators, and general public. Learners were exposed to the instruments that they wouldn't be exposed in the classroom if it wasn't for SMCRI. For instance, learners were able to use microscopes, laptops, and even boats that are supplied by SMCRI. This has had made huge contribution in their development of the ocean science understanding. As the result, more of the learners specifically are more ocean literate than before. For the general public, one of the things that SMCRI has provided us was the exhibition where learners and the general public were able to go and interact with the material that has been provided by SMCRI. Thank you.
My name is Dr. Sean Daisel. I'm a plankton ecologist working for SCION as the Research and Data Coordinator LTL1 Linode. I am also a research associate with the Institute for Coastal and Marine Research at the Nelson Mandela University. My research interests lie in estuarine and coastal zooplankton ecology, oceanography, ocean observation science, and LTR and information systems. Plankton as a field of study demands access to vessels, highly skilled staff and technicians and taxonomic specialists, and specific equipment to examine microscopic details of tiny organisms, including microscopes, very high-end microscopes in some cases. Availability of all these elements are rarely seen at any one university, department, or research team. Research infrastructure put in place by SMCRI over the past 5 years fills this gap in a significant way. Not only has it been instrumental in establishing vital regional and international research partnerships, opportunity for new research projects, and student recruitment into this field, it has also been vital in structuring our thinking and strategizing about the future of coastal plankton research in South Africa. With the foundation laid by the ongoing pelagic ecosystem long-term ecological research program underway in our Goa Bay Sentinel site, the success of which is highly dependent on SMCRI, we have been able to show the value to both research and human capital development that the local long-term plankton observation initiative can offer. Now, with the national rollout of research infrastructure at Sentinel sites along the KwaZulu-Natal and Western Cape coasts, we for the first time have the means to collaboratively think about and implement a national plankton observation strategy for South Africa. Such developments are in line with global efforts to study long-term changes in coastal ecosystems with the express aim of understanding the implications and opportunities for humans. From this perspective, the point I would like to leave you with is this. Without the present access to research infrastructure and future value proposition of SMCRI, our national contribution to such global goals will be limited. Thank you.
Hi, my name is Pumle Tojane Pondo. I'm an early career scientist within the L1 Linode as a plankton ecologist. SMCRI has enabled me to go through my PhD, which focuses on benthic diatoms, and with the microscopes that we have under the SMCRI program, the project has been a success.
Hi, I am Dr. Mfundo Bizani. I work for SMCRI as an early career scientist in plankton ecology. My research focuses on understanding zooplankton dynamics in relation to environmental drivers.
I'm Janine Adams. I'm Research Chair in Shallow Water Ecosystems at Nelson Mandela University, also affiliated with the Department of Botany, and I'm Deputy Director of the Institute for Coastal and Marine Research. And we've been using many of the SMCRI platforms over the years. We've seen research develop. A lot of our collaborative research would not be able to be done without these platforms. And what they do is they offer students activities, infrastructure for them to complete their research. And we have been particularly working with the biogeochemistry laboratory on water quality assessments of estuaries. This will include nutrients, looking at eutrophication, as well as metal analysis. And we have new equipment there provided by SMCRI that allows students to work with cutting-edge technology. And so this is very important for training of postgraduate students. The aerial platform is also very important. This is a developing one. But in estuaries around the coast, we're very interested in what the present health status of these systems are. And so we use aerial photography for mapping changes over time. And the platforms provide both aerial surveys. New technology that's going to be used is LiDAR surveys, so that we can look at elevation and responses of estuarine habitats to sea level rise and changes such as that.
My name is Rosemary Dorrington. I'm DST NRF Saatchi Chair in Marine Natural Products Research at Rhodes University, and we work directly in the field of marine drug discovery. So my group is interested in particular in bioactive compounds from marine sponges and ascidians and their associated microbes. And I lead a group at the moment that's focused on the development of antibacterial antibiotics. And we're also looking at using these compounds for antiviral drugs. So the work that we do is absolutely dependent on our ability to have access to field work and field studies. Our main field work is done in Algoa Bay, which is exceptionally rich in biodiversity, in the kind of biodiversity we're looking for. And so we would not be able to do that without the support of the SMCRI infrastructure, particularly the coastal craft that I have been fortunate to be able to use as a fellow SIAD for some time. We're also dependent on the remote remote imagery capacity. The remote operating vehicle is really important for our collection and sustainable collection of sponges and ascidians. And without the support of the technical staff and the crew of the ship, of the boat, we would not be able to do that work. Then I'm also involved in collaboration with researchers at SCION who have access to their biogeochemistry analysis facility. And a lot of our water nutrient sampling goes through the facility. We also have access to the data platform, the data management platform, that gives us really important metadata on the environmental conditions that accompany our sampling efforts, because that metadata is often critical in understanding the types of secondary metabolites that our sponges and ascidians and microbes produce.
Hi, I'm PhD candidate Rosalind Gibb, and I study bacteria and phytoplankton in the Gullis Current and Algoa Bay. We want to know who's there, what drives their distributions. Using molecular analyses and SMCRI resources such as boats and environmental variables measured through instruments that are in the sea and the microscope lab, I was able to identify the phytoplankton in these environments, as well as identify the drivers of their distribution.
Hi, my name is Shirley Parkenans, and I'm the Shallow Marine and Coastal Research Infrastructure, benthic Ecosystem Long-Term Ecological Research Coordinator here at SCION L1 Lake Coastal Node. Here we collect data on the benthic biota that is found on or close to the seafloor. My research focuses on shallow marine benthic ecosystems, and with a background in ascidian and sponge taxonomy, I am able to focus on both ecosystem and species. Working closely with the South African Institute for Aquatic Biodiversity's National Collection Facility in curating, identifying and describing new species, I am able to apply this information and use images of the benthic environment to describe community structure and biodiversity. And so we are able to get a better understanding of the effects of global change and the response of the marine benthic biota to these stressors. Samples collected with jump camera systems and stereo-baited underwater video systems allow for the collection of data without damaging the environment. It allows us to collect data from the same site over many years. Changes in the population and community structure of the benthic invertebrates and algae are echoed in changes in benthic fish populations. Fish is an important resource to many South Africans. However, disentangling natural and anthropogenic drivers of change is difficult, and for this, we turn to marine protected areas where human impact is minimal or regulated. The shallow marine and coastal infrastructure developed and maintained by SAION and SIAP allows us to observe change in different focal areas, from estuaries to the water column and the seafloor.
I'm Dr. Pagamanodo, and my research focuses on necessary habitats for fish in coastal areas. Thanks to SMCRI that I managed to collect my data for my PhD, thanks to Coastal Craft platforms and the lab equipment. Thank you.
I'm Dr. Carla Edrely, and my research focuses on the impacts of ocean acidification on marine organisms. And the most important component of this type of research is understanding local conditions. So the SMCRI platform gave me the opportunity to access infrastructure like boats and data to collect the relevant information for my study during my PhD.
My name is Dr. Polisa Jomo. I'm a physical oceanographer. I'm currently a postdoctoral research fellow at SAION L1 Node. My area of research is coastal oceanographic research. I look into the dynamics of the two bays, which is Algoa Bay and St. Francis Bay. So I use the research platforms, the data management platform, the coastal monitoring platform. Within the coastal monitoring platform, I basically use the UHRs, which is underwater temperature recorders, the ADCPs, which is the current profilers, and many other instruments.
Hello, I'm Dr. Edeline Bauer of the biohyperbaric medicine practice. We make use of the hyperbaric platform and the multi-place chamber to provide treatment for patients with medical problems like diabetic ulcers on their feet and crash injuries, hyperbaric oxygen therapy, as well as diving related injuries. We are on standby for the diving community of